From our chapter on blood, we know that the master stem cell creating all of our white blood cells and red blood cells and that megakaryocyte there is called a hematopoietic stem cell. This stem cell is capable of dividing into two other quasi-type stem cells that can differentiate into the final cells that we see in tissue and blood. We have before discussed the myeloid stem cell that gives rise to the erythrocytes and 4 out of 5 leukocytes and platelets, as I mentioned. We've talked about macrophages, or cells that eat debris, as well as dendritic cells, which are cells that drink fluids in order to ingest debris. This is called penocytosis. Both macrophages and dendritic cells derive from monocytes that are floating around in the blood and then decide to get out and take residence in a certain tissue. What we haven't previously discussed is a lymphoid stem cell, which is capable of dividing into T cells, B cells, and natural killer cells, which are abbreviated NK cells. T cells can divide into a whole bunch of versions of T cells, two of which we will consider. And B cells can also divide into plasma cells, which are cells that pump out antibodies, officially called immunoglobulins. The term phagocyte just refers to cells that eat things. More specifically, there are macrophages derived from monocytes, and then there are microphages. Macrophages are neutrophils and eosinophils, and they circulate in blood, and they're only summoned into a tissue when they're needed. You can see this neutrophil here is phagocytosing some bacteria and some other foreign cell type. When phagocytes encounter a pathogen or debris, they surround it with their cell membrane and then they ingest it into a small compartment called a phagosome. This was a process we saw earlier. It's a process called endocytosis. The phagosome then fuses with a lysosome, and that lysosome contains enzymes that can break down and digest the material. Special to only macrophages and dendritic cells is the ability to present these antigens of the pathogen to B and T cells in the lymph nodes saying, hey, I found this, I don't know what it is, but maybe we should be worried. This is a process called antigen presentation, and it's discussed in another mini lecture. Monocytes are white blood cells that derive from the myeloid stem cell. Monocytes, they float around in your blood. However, they don't spend the entirety of their life in blood circulation. They'll move into tissues in the body and take up permanent residence. Once there, we usually give them a more specific name that implies the tissue. You might not have realized this, but osteoclasts, those were cells that were capable of breaking down bone. They were not derived from other stem cells that give rise to bone cells like osteoblasts and osteocytes. Osteoclasts they're actually a monocyte that has moved into bone and taken up permanent residence. In the brain, you have microglia, part of the neuroglia, and in liver cells, there are cells that are called Kupffer cells. In your lungs, there are cells that are called alveolar macrophages. There are there's many different macrophages in the spleen, not just the four of them that you see that are listed here. Mast cells are important to us in this class. Mast cells reside in tissues instead of floating around in the blood. Mast cells release histamine and heparin in response to encounters with pathogens. Histamine induces vasodilation, and it has the ability of drawing more immune cells to the site of injury. In addition, histamine's effect on slowing the spread of pathogens at the infection site is primarily due to capillary plasma leakage or exudate and clotting proteins leaking into the tissue spaces. So, therefore, the greater the concentration of histamine released, the greater the inflammatory response. And I just want to make a point that anaphylaxis is the greatest concentration of histamine that you could release. So, mediation of inflammation through complement, those complement proteins, it serves to temporarily repair the tissue and it prevents additional pathogens from entering the wound. But it also slows the spread of pathogens from the site of injury or infection. So, let's say that your skin becomes wounded, like it is here in this picture. And whatever it is that punctured your skin was covered in little tiny pill-like bacteria, which are called bacilli. The first thing that happens is that resident macrophages that live in your dermis, they swallow one of these bacteria. And the macrophage releases all kinds of chemicals called cytokines, or more specifically, interleukins. The cytokines diffuse away from the macrophage and they enter into nearby capillaries in the subpapillary or cutaneous plexuses of the dermis of skin. Once in the endothelium, the cytokines are displayed on specific receptors sticking off the endothelial cells into blood circulation. And so this is how it works. White blood cells that are like flowing by in the blood, like neutrophils or eosinophils, they sense the signal of the cytokine and they start to roll along the outside of the epithelium, hoping that it'll be able to attach to the receptor with the cytokine that's being displayed. So when the attachment happens, the endothelial cells will dilate and they allow just enough space for the white blood cell to squeeze itself between the endothelial cells and into the surrounding tissue. And this is called extravasation, or it can also be called marginalization, but marginalization just refers to the dilation of the capillary, whereas extravasation refers to the migration of the neutrophil into the tissue. All right, regardless. So now the white blood cell, the neutrophil, it's drawn toward the injury site and it performs something called chemotaxis, which is just movement toward a specific stimulus. White blood cells are capable of something called diapedesis, which is a moving process wherein they disassemble their cytoplasm and then like reassemble it to move forward. I'm sorry, they disassemble their cytoskeleton and reassemble it to move forward. Once at the injury site, the neutrophil can then begin to do phagocytosis itself. It can consume the pathogen. It can release its own cytokines to recruit more white blood cells to come to the wound, and this starts like a positive feedback in terms of signaling more cells to come to the wound. Natural killer cells don't require to know what it is that they are killing. It's this anonymity that makes them part of the immune system. So natural killer cells, they go around and they are always frisking the cells of your body and confirming that the cells are wearing your identification tag. If that identification tag is not put on right or spelled wrong, a natural killer cell will just phagocytose whatever it finds without regard for its identity. It doesn't care. Unlike phagocytes that engulf the entire prey, a natural killer cell does this by attaching itself to a target cell membrane, and then it secretes an enzyme called perforin. Perforin assembles itself in the cell membrane of the pathogen, and it makes a hole in the cell. So at this point, the cell could possibly die from its contents spewing out, but that's not enough. The NK cell is the natural killer cell is like, I'm not going to wait for that to happen. And the natural killer cell secretes granzymes into the target cell. These granzymes are like little digestive stomachs, and they basically digest the target cell from the inside out. Once the target cell is pretty well consumed by the granzymes, a natural killer cell will then phagocytose the leftover debris. NK cells can also secrete cytokines that kind of act like flare guns to augment the inflammatory response. The cytokines would draw other white blood cells to the area. This type of attack is very similar to how another lymphocyte works that we will talk about in a later mini lecture. And these are called CD8 T cells or cytotoxic T cells.